Okay, so we are live now. Hello, everyone. I hope you can hear us. I hope you can see us. Please let us know if you can hear us and see us. There's always a bit of delay, but we will wait for people to say something in the chat to make sure that we are talking to people instead of to ourselves. Uh, yeah, let's wait a bit. In the meantime, maybe I can start sharing my screen. I didn't test my. Yes, we can hear. Okay. Yeah, so we are very glad to welcome you all to this fifth session of the Reading Club. We are very excited. We have Maria Tosches and Loreta Medina as invited speakers. And of course, we have Luiz Puelles and Paul Chisek as usual speakers, commentators. And yeah, what I will do is just to share my screen because we have a lot to talk about and not much time. So I need to. Yeah, so Alex left because I need to share my screen. Okay, so here's my screen. I hope you can see it. Okay, this is my last slide. So I will start the presentation. Please confirm that you can see the slide. Can you see it? I cannot see you, so you need to tell me. Okay.
Yes, it's good.
Okay, so as I was saying, welcome you all to this fifth session of the Brains Through Time Reading Club. We are going to talk about The Conquest of Land by amniotes and reptiles. Sorry, I need to do something. Can you mute yourself because I'm hearing an echo? I don't know if it's who of you. I think it's Luis. Luis, I will mute you, okay? Yeah. Otherwise, I'm hearing myself. Sorry for this. Okay, so as I was saying, we will be discussing this Conquest of Land chapter. We will be talking about early amniotes and sauropsids, the age of reptiles, as the chapter is called. This is the fifth chapter of the book. We have learned a lot in the previous 4 chapters, and we still have a lot to learn. We still need to talk about mammals. So yeah, we are very excited. But today, we will be talking about amniote origins and the age of reptiles. Let me just briefly mention who are we, who is the Reading Club. It is Raquel and Alex. We are in the background helping in the chat and answering your questions. Myself, we actually take care of the logistics of the Reading Club. And then we have Luis Puelles and Paul Chisek, as I said, as usual commentators that provide the main content of the Reading Club with their knowledge about the topics we discuss. And today we are very excited to have Loreta Medina and Maria Tosches, who will be telling us about different topics that are relevant for the chapter we are discussing today. We have Loreta telling us about the homologies and convergence of sauropsid Pallium, and Maria Tosches will be talking about this cell type profiling approach and what we can learn and we cannot learn about from this technique. But before that, I will try to do a summary of the chapter. It is a very long chapter with many ideas. So I will skip a great part of the brain information, the information about the changes in the brain, because Loreta and Maria will talk about those changes much more in detail. And my main goal now is to give you an idea of the context in which this brain was evolved. Okay, so I will. Other thing I want to mention is that I reorganized a bit the figures because it felt a bit more natural to me, but I indicate which figure I'm talking about in each slide, so hopefully you can follow easily. Let me just briefly mention the previous session in which we talked about the Lys amphibia with anurans, urodels, and caecilians, which had a relatively developed brain, which is helpful to compare to the brains we are discussing today, that are the brains of sauropsids that are higher in this tree. So this is this branch here, the amniota branch, that actually contains the sauropsids, that are the ones that we care today about, and the synapses. I don't know if it's synapses or synapses, but that will give rise to mammals that we'll be discussing in chapter 6. Okay, so basically our sauropsids emerged around late Carboniferous, and this animals were happily living in this environment that you can see in this beautiful picture that you can find in the book. So they were eating insects that already had conquered land, and they were living between water and land. But at some point, actually through millions of years, something happened that made life of amphibians a bit more difficult. There is this collision between Laurasia and Gondwana continents that created this Pangea continent, huge continent, which reduced actually the coastal habitats and produced a more continental environment. And so when with this environment, with these conditions, it became kind of a good idea to develop innovations to be able to survive in drier conditions. And this is what Amniota did. Amniota developed this amniot egg, which is more resistant to dry environments. Their skin was covered by scales that makes the skins more resistant, and they develop a more complex respiratory system with larger lungs and trunk muscles that allow pumping in and out the air of these lungs. This complex respiratory system was necessary because the scales may impede the gas exchange through the skin that we have seen in amphibia. So this is our amniota, and within the amniota, as I said, we care today about the sauropsids that are all these families that I will give you some details now about. So we start with the Tuatara, that is the most basal squamate, is this cute animal with a relatively developed brain that has this olfactory bulb connected through these peduncles to the rest of the cortex. The Tuatara is the closest, or has the closest relative, the Lepidosaurs, this lizard, as I'm showing here, that has a relatively similar brain with the peduncles, and in size is very similar, as we will see later in this presentation. And when we move to turtles, we go up and we go here. We see that the brain of the turtles is a bit different, with the telencephalon that occupies a relatively larger space area, and with the olfactory bulb that is closer to this telencephalon. Turtles were actually thought to be the most basal sauropsid and at some point, but recent studies have shown that this was not the case. And you can see the branch here leading to turtles. We move up to archosaurs that have crocodilians as one important group. Crocodilian's brain is similar to that of turtles, but have again this pedunculus because the olfactory bulb is at the tip of this snout, the typical snout of crocodiles. And finally, when we go to the top, we find the birds that are the ones that have the largest brains. You can see that the telencephalon is actually much larger in proportion and in absolute terms, and the cerebellum, as we will see later, is also very well developed. You can see also the olfactory bulb is relatively smaller in comparison to the rest of sauropsids. Okay, so birds, as we all know, are the descendant of dinosaurs, and I didn't want to forget about dinosaurs. One of the most famous is the Tyrannosaurus rex, that is this angry animal that along the branch that leads to birds became more smaller and smaller in brain size and also in body size. And you can see that this theropod developed these feathers and became smaller and smaller. And here we can see this Archaeopteryx, that is a close relative to modern birds. Okay, so these are let me just go back. So these are our main protagonists of today's session, which actually had to develop or adapt their senses to the new habitats they were conquering. In vision, for instance, turtles and lizards which live close to the ground develop this horizontal strike that allows them to perceive in focus objects that are on the ground, along the ground. And this is what they say: when a turtle looks straight ahead, objects on the ground will tend to appear in focus regardless of their distance. And these red dots is the focus at each distance, as you can see. Another feature that seems to be deriving in sauropsids is this fovea, that is a thinning of the retina that allows to reduce light scattering and thereby improving visual acuity. And you can see the steep walls of the tuatara fovea, which they propose that might help bending the incoming rays of light, increasing the visual resolution in this small part of the central visual system. Okay, so this is regarding vision. Then we move to hearing. Hearing in land, it becomes very important to perceive or to be able to detect high frequency sounds, because you can maybe detect flying insects, because you can hear mates vocalizing, or and this is especially important for owls, for instance, to locate sounds with a high precision. And to hear these high frequency sounds, different sauropsids develop different mechanisms. For instance, lizards apparently increase the number of cochlear hair cells and this helps them hear sound frequencies higher than 1 kilohertz, while the Archosaurus, what they did is to make larger this cochlear duct that has been shown to correlate with the range of frequencies that the animal can perceive and can hear. And you can see here the barn owl, that is actually, it seems to be one of the animal that is able to perceive higher frequencies, and this is important for them because they need to locate animals in the dark. Okay, in olfaction, they mentioned several features. I found interesting that sauropsids develop different olfactory receptors depending on their necessities. And one of the most interesting things that I found is that I read is that seabirds are able to detect dimethyl sulfide that allows them to detect large schools of fish that they can then hunt. And reading about this in Google, I actually found another news that is a bit more recent, that apparently plastic in the ocean also has this dimethyl sulfide, and that's why seabirds are eating so much plastic and they are dying because of that. So this is a bad joke of evolution. Okay, so let's move to changes in motor patterns and control. So one important thing is that, especially in Archosaurs, the animals, dinosaurs and birds became erect, so they stand on their limbs, which helps them have a higher visual point of view to detect prey and to detect predators, and also to save energy, because part of the weight is put on the limbs instead of. So this other stance seems a bit more tiring, right? Or at least seems intuitive to me. You can see actually that crocodiles have this type of walking that I didn't know about, that they can have this semi-erect stance. And regarding respiratory system, I already mentioned that they develop these trunk muscles that allows them to pump air in and out. But it seems that lizards didn't improve entirely their respiratory systems, because apparently they cannot run and breathe deeply at the same time, because when they run, they move sideways and the air from one lung goes to the other. And you can see actually this figure kind of proves this, because when they are not running, there is airflow at nostrils, but when they start running, there is no airflow. So it's like they hold their breath when they start running, which doesn't seem very efficient, but yeah, they don't mention more about this. Okay, so let's move to the brains, to the changes in the brain. I don't know how much time I'm using. I want to spend some time in the size of the brain because there are several figures that seem relevant. I think there are 3 figures in the chapter talking about the size of the brain. The first one is this figure 4, in which we compare the size of the brain of non-avian sauropsids, and you can see. I don't know if this is something that I deduced, but that I infer. But it seems that the brain of lizards and snakes was not much larger than that of amphibians, in this case anorans. You can see the dashed line. But one thing that is true, it seems that lizards have a relatively a bit larger brain and larger than snakes, which seems to have a smaller brain relative to body size. But it is when we move to birds that things become much, much larger. You can see here that almost, well, maybe all birds have larger brains than reptiles. You can see here the dashed line that indicates the size of the brain of reptiles. And it is the parrots and the ravens and the perching birds that have the largest brains. And they say that an average avian brain is roughly 10 times large as the brain of any other sauropsid. So birds are smart. This is one take-home message. Another study they show is this comparison between different species, and you can see that the size of the brain of parrots and corvids is significantly larger than the others. And the other animals, among which there is the Tyrannosaurus rex that is very upset because it doesn't have a large brain. And finally, regarding the structure of the brain, the main structure, they also mention at the beginning of the chapter that the telencephalon, the percentage taken by the telencephalon grows. So as we move from amphibia, you can see 20% in bullfrog, to tuatara, and when we reach chicken, we have 50% occupied by the telencephalon. And actually, they mentioned that in parrots and corvids, this reaches the 80% of the total brain. So telencephalon becomes the most important area in birds. And of course, this is also true in mammals. Okay, regarding the different parts of the brain and the changes that happened, I will use this figure that is from chapter 2, just to highlight the region we are in. So regarding the hindbrain, they say that it didn't change much in structure, but some important innovations arise, like this delay line that allows birds to detect interaural difference. So this difference in axonal length helps the neurons to detect sounds that come from the contralateral or ipsilateral or somewhere in between, depending on where the sound is. One of these neurons will fire. If we move to the cerebellum of birds became very large, as I mentioned before. And one important, interesting aspect is that it became very similar to, or it developed in a very similar way to that of mammals. One of the similarities is this folding structure that is very typical of mammals' cerebellum. So the cerebellum in birds apparently became larger because of this erect stance that one of the reasons, at least, is this erect stance that I mentioned before. Then in the midbrain of sauropsids, the most important structure is the optic tectum. Here you can see a cut of the optic tectum of a turtle. This optic tectum is organized in 2 layers. The most superficial layer receives retinal axons or retinal inputs, as you can see here, and the deepest ones receive somatosensory and auditory input. So regarding this retinal input, you can see here a more detailed schematic in which you can see the retinal axons connecting to the dendrites of the tectile ganglion cells. So the most important function of optic tectum is to decide where to pay attention to. And this is done via a winner-take-all competition in which this tectoismal pathway is contains these inhibitory magnocellular neurons that are GABAergic that will inhibit all neurons but the ones that are active. So as soon as one gets active, this will activate these inhibitory neurons that will inactivate all others, and thus these guys will be the ones determining which part of the visual field to pay attention to. Okay, so regarding the telencephalon, as I mentioned before, Loreta and Maria will tell us much more. I really like this figure in which they show the divergence of the telencephalon development between sauropsids and mammals. You can see that in the sauropsids, the ventral pallium became very large and produced this dorsal ventricular ridge, and the superficial part is the olfactory cortex. And in the mammals, as you might already know, it is the dorsal pallium that enlarges considerably, becoming the neocortex. Regarding this dorsal ventricular ridge, it can be divided in two different regions, the anterior and the posterior, and it has been shown in birds that is separated into layers. You can see the different layers, and these layers are interconnected, which seem to be similar to the neocortex of mammals, but only superficially. And actually what they say is that it is the dorsal cortex of sauropsids that in birds is called the vulst, that seems to be homologous to the neocortex. And actually they show this figure showing that the owl's vulst is very similar to the primary visual cortex of mammals. So this is all I wanted to say. I know there is a lot of information that I didn't explain, but I think it's better if we let Loreta and Maria tell us about more in detail about the pallium of sauropsids and about all these details, and then we can all discuss and learn. That is what we are all about, why we are all here. If you want to discuss, remember that you can ask a question with this button here, and you can make comments in the chat. Hopefully you are already making comments. I cannot see them. And let me just thank Worldwide Neuro for letting us use the Crowdcast platform, as always, and the Barcin community in Barcelona and the Instituto de Neurociencias for their help. And I will actually, I think this is the end. We'll stop sharing and let Loreta talk. So maybe you can unmute yourself.
Yeah. Thank you, Manuel. Thank you for inviting me. Let me share my screen first. I think it is this. And let me go here. Now you see my screen, I guess.
Yes, go ahead.
Yeah, okay, good. So let me put also the pointer. So that you see better. No, you see the pointer now.
Yes.
Yeah. Okay. Good. So, well, as I said, as I said before, I thank Manuel and all the group that is organizing these very interesting sessions for inviting me. It is an honor to be here, and also in the company of Luiz, Maria, Paul. Also, so many people, including Georg Striedter, who is one of the authors of this very interesting book. So today I'm going to talk mainly about, well, or only about the Pallium. I'm not going to tell many details because I will just talk about a few things, and I will raise some questions that we can discuss later. And I will also leave some space to Maria, that she's also going to talk about a little bit about this. So why the Pallium? Because it's the most, or one of the most divergent parts in the telencephalon or in the brain of amniotes. One of the things that we need to understand or to discuss about when we talk about the Pallium is how many divisions are there and what was the ancestral condition. Many times people look at this and start looking at this, looking at the Pallium of mammals. When and they get a number of divisions, they try to find the same divisions in non-mammals. But this is not really very convenient because of the high divergence, and it is very important to analyze what happens in sauropsids. And sauropsids are also very divergent, and it is interesting then to look at different groups and different lineages, different paraphyletic groups, etc. And this is what I will try to do, to show several examples. We need to keep in mind that perhaps when looking at all of these different sauropsids in comparison to mammals, perhaps we might find out that not all divisions are there, and perhaps some divisions evolved later or were very tiny and were enlarged in particular lineages. So we also need to be aware that if we find homologous divisions in different species with different sizes, perhaps they, during development, might include some subsets of progenitors that are novel in some groups and are not present in others. So this is the basis for perhaps giving rise to new cell types. So when we find homologous divisions, not everything in different groups, not everything is going to be the same. Some things might be conserved, some things might diverge, no? Why it is interesting to identify these homologous divisions? Because it's a better way to differentiate between conserved patterns, divergent patterns, and also to identify some similarities that perhaps are not due to inheritance to the common from a common ancestor, but are due to convergence evolution. Understanding all of this is important if we want really to understand the organization of the brain, if we want to address better, more focused questions on brain organization and evolution, questions that are have a better more important significance, biological, no, that are biologically relevant, and also this by analyzing the brain like this and identifying correctly the homologous divisions, perhaps this can help us also to make predictions by looking at one of the division in one of the animals and looking at all the features that are found there. Perhaps we can go to a different animal and try to find some features that were unnoticed previously. So it's also interesting for making predictions. So as I say, many times we work with models, and many times models are produced by looking at mammals. And this is like, and then trying to find them in non-mammals. And these are three models that have been, there on the organization of the Pallium, from starting from the tripartite model, which consider the Pallium divided into three main divisions: medial, dorsal, lateral. The medial is the one that produces the hippocampal formation. The dorsal is the one that produces in mammals the neocortex. The lateral is the one that produces the olfactory cortex, in addition to other groups, including the endoperiform nuclei, etc. But More recently, Luiz and collaborators proposed a different model of 4 pallial divisions, the tetrapartite model, and this was based on analysis of the structure during embryonic development and taking advantage of analysis of gene expression patterns, in particular expression of developmental regulatory genes encoding transcription factors and other highly conserved proteins that play relevant roles in the development of these different divisions. And by analyzing that, they saw that this lateral region over here is really divided into two main subdivisions, two main divisions, and this took them to propose a 4-part model, a tetrapartite model, which has changed a little bit with time to accommodate new data. So now in the updated tetrapartite model, they consider the lateral pallium as that part of the pallium that produces the claustrum and the insular cortex, and the ventral pallium is below, producing the olfactory cortex and some endoperiform nuclei. And more recently, caudally, they also found an amygdalar pallium, which is different from the ventral pallium. In recent times, we also proposed another model, which is called the hexapartite model, which six pallial divisions. And why? Because we were trying to compare the pallium of a reptile, a lacertid lizard, with that of a chicken, with that of a mouse, and we found that dividing the pallium in 6 parts was easier, made the comparisons easier. It was closer to the divisions that we really saw. And for that, we also used a number of expression patterns that help us to identify and distinguish the divisions. But how is the pallium in sauropsids? This is an important question to really see if the models that we are proposing are really good as models of the amniote pallium. And I remind you, based on this simplistic scheme, that the sauropsids include the birds and the reptiles. You can refer to figure 5.1 from the book chapter, where you have a more detailed diagram, phylogenetic diagram, where you see the different groups. But basically here you have the birds. The crocodiles and like the alligator are closer to birds, and then you have other lineages producing the squamates like snakes and lizards. In a different line you have the turtles, etc., etc. So when we compare the brain of a pigeon with the brain of a rat in a frontal section, and stain just for using a seat architectonic stain like a Nissel stain, you can see that they look quite different, and it is very difficult to distinguish here where is the pallium in the pigeon. For that reason, during many years, many of the divisions in the pigeon, in the reptilian telencephalon, in the avian telencephalon in particular, had names suggesting that they were divisions of the striatum. No? The striatum is down here. In rats, the pallium is in this part over here. Large parts of the pallium are laminated, as you see here, like the neocortex, no? Like the olfactory cortex, etc. But here you don't see that. Only when researchers started to use and to incorporate data of histochemistry, immunohistochemistry, and also adding information on connections, etc., they realized that this lower part here that you see dark in this image on the top, right top, this part here is only this part is the subpallium, and the pallium is just all of this territory above. And you can see that it's quite different from the pallium in mammals. And it also, if you see the seat architecture, you see that it's divided into several divisions by these cell-poor laminae. So you have many of the divisions. These cell-poor laminae, you don't see them in the pallium of mammals. In mammals, you see some different cortical areas with different organization, but you don't see cell-poor laminae as you see here in birds. So one question is, why are these cell-poor laminae produced? And we can discuss this later. I have no idea, but it's something interesting to discuss. So this large territory here of the pallium of these animals is included as part of the dorsal ventricular ridge. But how is this in the classical reptiles? Well, first, this is from the atlas by Puelles and collaborators of the chicken atlas, and you can see the different cross sections through the pallium, through the telencephalon, from more frontal to more occipital levels, and you see the different divisions. This division here is the bulls, and here you have the DVR. You see here the olfactory bulb. In the DVR you see at least two divisions, and caudally you see an extra division, which is called the arcopallium, that you see here, at more caudoventral levels. And in this division that is called the nidopallium, this here that I'm showing you with the pointer, you have several specific nuclei, like this nucleus that vasorostralis, this nucleus that is the antopallium, and this other nucleus that is the Phil L, which are different sensory related nuclei of the nidopallium, receiving distinct types of sensory information. How is this in non-avian sauropsids? And here I show you images from the egeconid lizard, the lizard gecko, also in frontal sections, and a turtle, the eastern painted turtle, Chrysemis, from atlases published by Smith or by Powers and Reiner. And you see here, this is the paleo-subpalliar boundary, and above it you have the DVR in the lizard, and you have the DVR in the turtle. In the turtle, it is quite prominent. It bulges on the ventricle, and this is the name comes from that. And above this, you have medial and dorsal to the ventricle, you have several cortical areas. You see them here in the lizard, and the idea always have been, where is the primordium of the mammalian neocortex here in reptiles, in classical reptiles. And some authors have proposed that the area that is called the dorsal cortex, is this area here or this area here, is that primordium of the is the primordium of the neocortex or of mammals. And also some authors have also included in this proposal the structure that is called the pallial thickening that I'm pointing here, this area here. And even some authors have said that in the dorsal ventricular ridge, you have cell groups that are comparable and even homologous to cell groups that you find in some layers of the neocortex of mammals. So there has been a lot of controversy when trying to find pallial divisions and pallial cell types in the pallium of sauropsid. And one way to look at this is by analyzing the brain throughout development, considering the topological position of pallial divisions, the relative position of these pallial divisions, and more recently, it has been very important to include gene architecture analysis, in particular, comparative embryonic gene architecture that help to identify the same divisions, the same divisions across different species. And when doing that, for example, this is related to the medial pallium. In the medial pallium, during development, this division is characterized by expression of the transcription factor LEF1, you see here, and also other regulatory genes like LHX9 or the link-only gene LMO4. When looking at these expression patterns in the embryonic pallium of the chicken embryo, you can see that the progenitor sum of the medial and dorsal plate, cortical plate, is expressing LEF1, and also this territory expresses LMO4 and even LHX9. This means that this territory, medial and dorsal, is comparable to the medial pallium of mammals. This is from a lizard brain, also an embryo. This is an intermediate stage, and you see expression of LHX9, and you see this dorsal cortex here with different subdivisions, and which are comparable to the two subdivisions that you find in a similar position, in an identical topological position in the chicken embryo. This means that this cortical plate here does not belong to the dorsal pallium but to the medial pallium, and it belongs to the hippocampal complex. No? It includes these parts over here, include areas that are comparable to the entorhinal cortex. But if the dorsal cortex of reptiles does not belong to the dorsal pallium but to the medial pallium, where is the dorsal pallium in sauropsids? In bears, like chicken, it is this structure that is called the bulls that protrudes dorsally here on the top, and that's why it receives this name, which is a German term that means something like bulls, and referring to this protrusion on the top. And where is this same territory in non-avian sauropsids? When trying to think about this, we were using genes like EMX1 and other several genes, not many, because we didn't have that many, or we didn't have the possibility of analyzing the messenger RNA of many genes in reptiles, but we look at a few. But we also pay very close attention to the topological position, and we try to find a similar territory here in this dorsal aspect, and in the frontal part of the pallium, above the DVR. Looking at this is from a lacertid lizard embryo, and this is from an agamid lizard. We found a territory that we propose could be the same, and at least it is in the identical topological position, and it expresses EMS1, and very importantly, it does not express LHX9, because one characteristic of the dorsal pallium in bears and mammals is is that it does not express LHX9. So this territory here, pointing with an arrow, is the area, and is also here, is the area that we propose that could be the dorsal pallium. And you can see, well, of course, this needs to be evaluated, analyzing perhaps transcriptome at single cell level, as Maria is usually doing in her studies. But one important thing here to consider is that this dorsal pallium in some sauropsids is very, very tiny, very small. Perhaps in other sauropsids, well, like turtles and like crocodiles, it might be a bit larger, more resembling the situation inverse. But this is something that needs to be analyzed in the future. And one thing that also needs to be considered is how was the evolution of this dorsal pallium? Perhaps it evolved independently and it became large independently in different lineages. It's something that needs to be analyzed in the future. So regarding this lateroventral parts of the pallium, is the region that includes the dorsoventricular ridge, at least according to our hexapartite model, and is a territory that is characterized by expression among several other genes, the link-only gene LMO3, and you can see here that it includes, well, 3 different divisions. This is in chicken. It contains the nidopallium, which is the ventral pallium. It includes the mesopallium, which is the lateral pallium, and caudally, it also includes another division that is the arcopallium, which is part of the ventrocaudal pallium, as we will mention later. So these divisions, where are this territory expressing LMO3 in mammals? And this is an embryo of a mouse, and you see the territory expressing LMO3. Basically, it includes the piriform lobe, as it is mentioned by several authors, including Shubatol and her colleagues. It includes the piriform cortex and the endopiriform nuclei, and caudally some parts of the pallial amygdala. So above this domain, you have the claustroinsular region. And where is the claustroinsular region in chicken? Is, according to our view, is here above. And you can see here that we have some discrepancies in the interpretation of these territories, especially the comparison between mammals and sauropsids. Some discrepancies here with Luiz, and he knows, excuse me, Luiz, we can talk about this later, but this is how we understand the pallium now. So we have called this territory above the dorsolateral pallium. This territory above expresses cadherin 8, exactly as it does the claustroinsular region in mouse. Is the DVR, does the DVR contains these 3 divisions found in the chicken or another birds? And the answer is yes, it appears that it does. Here we have the case of an alligator from an article by Briscoe and colleagues, and here they have in the DVR a ventral sector and a dorsal sector with a molecular profile very similar to the corresponding divisions in the chicken and another birds. And caudally, you have also another division that would be comparable to the arcopallium of birds. So yes, there are 3 divisions, not only in birds and crocodiles, but apparently also in squamates. At least this seems to be the situation in lizards. The only thing that in lizards this is smaller, is a division Intercalated here between the dorsolateral pallium and the ventral pallium is this area here, pointed with an arrow. And here you have the same division. This would be like the mesopallium in Anagamid lisa, the tawny dragon. And finally, regarding the ventrocaudal pallium, is the very caudal part of the DVR, the dorsal ventricular ridge. You see is quite large or relatively large in birds, especially in some birds like chicken. Is separated from the rest of the DVR by this cell-poor lamina. You see here. It is very rich in genes like LHX9. And you have just a division in an unidentical position in lizards and other reptiles. In lizards and other reptiles, it produces some structures, including one nucleus that is called nucleus sphericus, which is specific of reptiles, and in particular is very large in some squamates, like snakes and some lizards, that have a very highly developed vomeronasal system. This nucleus sphericus, receives a prominent input from this system, and then projects is connected with other parts of the DVR and projects to the hypothalamus. So this is and other reptiles that with a less or a lower development in this vomeronasal system, they had a smaller nucleus sphericus. And in birds, like the chicken, where the vomeronasal system is tremendously reduced and perhaps absent, as many people claim, you don't have a nucleus sphericus, but the arcopallium develops other types of nuclei related to other functions and have undergone independent evolution. So where is this division in mammals? And this is the final thing. For that, it is interesting to look at this in sagittal sections. This is from chicken. Horizontal sections also from chicken. This is the arcopallium, this posterior part of the DVR. And if you look at this diagram from Luiz and collaborators like Elena Garcia Calero and other colleagues, you see the amygdala also occupying a similar position to the arcopallium, but this is more complex than this because the pallial amygdala includes, as they propose, a number of different radial units, and the arcopallium is not comparable to all these pallial units, but only the most posterior one that produces this gray area here that produces, among other structures, a nucleus. Sorry, it's not the nucleus, but a cortical area, the posteromedial cortical area of the amygdala that also receives vomeronasal input in rodents where the vomeronasal system is quite well developed. And where is the rest of the pallial amygdala? In other parts of the DVR, and it is unclear how extensive is this. Perhaps it includes the posterior parts of the anterior DVR and even intermediate parts of the anterior DVR. So this is something still under discussion. And this is all. Let me escape. I'm sorry because I think I took too much time. And well, now is the time for Maria, I guess. I closed.
So we actually will give some time for questions regarding what you told us. That is a lot and very interesting. So basically we have a question from the audience, but I would like to ask the other panelists if they want to comment on something or if they want to ask a question, feel free.
I would just say that question that Luiz Pessoa asked would be even better after Maria's presentation, I think, because it'd be great to have the bigger picture, all the different pieces. So maybe we can wait with that one.
Okay.
Okay, I didn't see the question, but it is okay to wait until later, of course.
Well, it was about the palliamygdala,
Okay.
which and the difficulty of finding it in the homologue. All that it's such a great question.
We talk a lot about this with Luiz, of course, Pessoa, and of course with Luiz Polles as well. And there is a very intriguing area which is called the inverse Pallium caudolaterale, that many people always compare with the prefrontal cortex, and it plays important roles in cognition, in decision making, and so on. But this is also the same for some parts of the palliamygdala. So, in fact, these parts of the palliamygdala are reciprocally connected with those parts of the prefrontal cortex. So they are part of the same functional system. And another way of looking at this is that makes more sense, perhaps, from an homology point of view, is considering this caudolateral part of the midopallium, in addition to other parts, as part of the palliamygdala, in addition to the arcopallium and in addition to other areas. At least this is my view.
If I may, can you hear me well?
Of course, yes.
I have a question for Loreta and Luiz, because I'm interested in their thoughts about this issue of the variable size of the dorsal pallium, as redefined and more precisely defined by Loreta in sauropsids, and this idea that parts of the dorsal pallium might have evolved maybe independently or expanded independently in sauropsids and mammals. And thinking also about the last chapter, where Luiz gave a very nice overview of thalamocortical connectivity, I wanted to ask about an update on thalamocortical connectivity, and particularly this issue of the presence of an LGN and of a visual cortex in sauropsids, because to my understanding, there is, part of the dorsal pallium of turtles, for example, is defined as a visual cortex because it receives this prominent LGN inputs. But there is nothing, at least in the literature, there is nothing comparable described in lizards, as in squamates, as far as I remember, but I might misremember.
Yes, that is a very important question, very interesting. In fact, Luiz, also, in 2001, Luiz, you wrote a very interesting article about this, talking about the thalamo-pallial connectivity. And he clearly explained there that the projections from the thalamus that arrives to these sensory areas, the primary sensory areas in mammals, to the mammalian neocortex, come from lengthnothalamic nuclei, while the projections that are the nuclei, the thalamic nuclei that produce the projections and provide the sensory input to the region of the DVR and the amygdala, come from colothalamic nuclei. These are in different embryonic tiers, different embryonic divisions of the thalamus. And Luiz explained this very, very nicely, well, in several of his articles. And yes, I'm going to the other question. I leave space to Luiz if he wants to comment about this. But talking about the other thing is, yes, in lizards, you don't have a projection from the dorsal geniculate nucleus reaching an area like the true dorsal pallium. It arrives to the pallial thickening, but the pallial thickening is a different story. The pallial thickening, I didn't talk much about it, but according to in our view, our group, we think that the pallial thickening includes several parts in different divisions: dorsolateral pallium, lateral pallium, even ventral pallium, because some authors provide a very large pallial thickening with different things there. So it's a structure that needs to be reanalyzed. and the connections also reanalyzed, but nothing reached the true dorsal pallium. Well, excuse me, 1 second. The true dorsal pallium in lizards is in the very frontal pole of the pallium. And in fact, this is something that needs to be studied. Nobody pay attention to that area, this tiny area. So the first thing that needs to be done is to analyze the connections of this very frontal pole to discard anything that perhaps was missed, because people started to look at the pallium a bit more caudal when you already have the different cortices that belong to the medial pallium. So it's something that needs to be studied in the future.
I had understood that at least in turtles there is a lemnothalamic projection
to the dorsal cortex, but
unless this nucleus has been mislabeled. But
yeah, but perhaps this is an it needs to be perhaps this is an independent evolution of an independent enlargement of the dorsal pallium in turtles, perhaps also in crocodiles, we don't know, and like in bears, you know, that you had the bulls, but not in squamates, or it's not something that needs to be reevaluated.
If I may say something,
Of course.
I think that the whole issue of connections in the sauropsidian telencephalon compared to the mammalian one or to anamnios and so on is very chaotic in general. The famous theory of Harvey Karting about his odological particular cell groups that might migrate or not migrate in mammals and so on, well, that has played havoc with the thinking about the matter during many years, because this hypothesis was published in '67, and since then nobody has been able to see order in the connections of the telencephalon. The problems are terrible because we have different nomenclatures, different concepts. We don't speak of the same thalamic nuclei. We don't speak of the same telencephalic areas. We don't look at the same sections. So it's absolutely dramatic how much harm this innocent hypothesis of Harvey Karting has introduced into the field. So I thought that in 2001 that the ideas of these parts of the thalamus could be introduced by Ann Butler were useful in order to clarify at least the thalamic part. And in fact, in our last session of the club, I presented some data about the amphibian thalamus and some aspect of the division of the thalamus and so on, which were in connection with this particular hypothesis. So I think that this is one line that merits more analysis and more molecular and causal analysis, because these parts of thalamus, I believe in them, let's say, at this moment. So I feel that all the data from the 70s up to now need to be done again. And for that, we first have to clarify also the topography in the telencephalon in the Pallium, because Loreta is speaking of 6 sectors of the Pallium, and speaking of 4. I am trying to move into a ring-shaped pattern of the Pallium. So again, this is very perverse to the clarifying of ideas. So I am completely aware that we are not at no place are we close to being clear about what is the situation. So new people in the field, like for instance Maria, if you are starting to do connections, then be advised about the difficulties, but try to clarify as much as you can with your own methods and your own approaches, because we really need new ideas in the field. We need new people, new approaches, and some sort of synthesis. We need to find some common arguments to what should be done. How should connections we compared among different vertebrates? It's not clear at all. Every time we try to discuss this sort of thing, the people don't agree.
And what you're saying, if you're saying that all the data from the 70s need to be redone, I don't know if it's good or bad news. You know, this is very hard. And
including the gene mappings. Because I have found I was erred in several of my earlier papers, as it was normal, because we were very innocent and we tried our best. My first paper on the genes in the Pallium, that was in 2000 or 99 and 2000. It took me 4 years. It took me 4 years of very deep thinking about what I should say or not say. And after 10 years later, I found I had committed several errors. So everybody committed several errors. So that's the problem. So me, you, and he, she, everybody. So the field is a bit chaotic. We have to admit that. And so the people that want to introduce order into this, the new generations, they will need to systematize, say who was right, who was wrong. Maybe every one of us did something right and committed some errors, so we have to dissect the situation and find a synthesis where so I can admit my own percentage of errors, because I already found myself several of them, so I may have some more. So I can admit that in some point where I discrepancy with Loreta, she is right and I am wrong. Why not? So I can admit it easily. So I have no problem but she needs to show me the evidence, of course. So, but one problem we have is that I have never seen her material, and I don't know how far she has seen my material. So since we separated physically, she moved to Catalonia and I remain in Murcia. We have not had so much time as we had before to discuss things and comment and so on. So I miss being close to Loreta and to be able to say, look at this, and I found this data, and what do you think of this or that? So we have lost a bit of that proximity, so it's a pity, but still I have to convince her and she needs to convince me. So maybe we are both wrong, and then it's some other solution that is involved. So it's a very difficult, everybody has feeling big difficulties in this particular part of the field, and I would suggest also the people working on the field of carton to try to assimilate all the weight of evidence coming from genetic work and from developmental work and from transcriptomics and so on, that is accumulating on top of the more classical evidence. And we cannot continue speaking only of connections. We have to deal also with sectors in the Pallium and boundaries and patterning, and we need to deal with all the modern concepts and try to make a common story. So that is my position. So if you ask me whether I understand the connection, I tell you no, I don't understand them.
So if I can stop you guys there, because Maria has to tell us about her work, and we can discuss later when she finishes. So Maria, if you want to share your screen.
Yes, I will share my slides in a minute or so. I first want to say I have only 2 or 3 points, so I really don't want to spend so much time in the presentation. But I really want to preface this by saying that this field, like any field in science, is being technology driven. And obviously all these discrepancies that Luiz and Loreta already alluded to were partly the result of the use of different, of the choice of different methods and different approaches to study the same problem. And indeed, the issue of DBR and cortex and homology or convergent evolution is one of the most outstanding examples of this situation, where scientists have looked at different parts of the brain, at the same parts of the brain with the different methods, and arrived at different conclusions. So now we're living in an age where transcriptomics has become much easier than what it was 5 or 10 years ago. We can profile single cells with the transcriptomics resolution. But I would still like to emphasize that this new technology should not override what we learned from the past, and that I think it should be the complement of all the knowledge that people like Luiz, Loreta, and others have collected in the last decades, because otherwise the risk is to get lost into this huge mass of data and to draw the wrong conclusions from these rich data sets. So I think instead these data sets are a good opportunity for us as a community to do exactly what Luiz said, to bridge the gaps between these different models and ideas on brain evolution. So let me share my screen. Okay, I apologize. It takes me a moment. Yes. And now, can you please tell me whether you see my presentation?
Yes. Yes.
Okay. So as laser point. So as I was saying, the problem of cortex and DVR is the prime example of the issue of what do we conclude if we start from different approaches. And so here I have I'm contrasting these two hypotheses, the equivalent circuit hypothesis that started with a landmark work by Harvey Carten, and then the tetrapartite Pallium model as an example of the many models that Loreta discussed on the developmental-based approach to compare brains across species. And so I'm not going to repeat what Loreta said, but the underlying idea is that this school of thought is that when we compare developing embryos, we can recognize regions that arise from similar topological positions that express similar sets of developmental transcription factors, and therefore we can infer potential homologies using these criteria. The connectivity equivalent circuits school of thought instead places more emphasis on connectivity and on in the case of the telencephalon, for example, it has been proposed that neurons that receive thalamic inputs are homologous, neurons that project outside of the pallium are homologous, and that these intratelencephalic neurons that connect these different layers of input and output processing are also homologous across species. And so this translates into a proposal of homology of neurons in the DVR and neurons in the mammalian neocortex. In the neocortex, these input and output and IT neurons are distributed in layers, and instead in the DVR, they are distributed in complex ways in nuclei. So the reason why this has become even more confusing in recent years is because molecular data have been used to support further these models. And so one will see in the literature papers where the use of certain genes supports the tetrapartite Pallium model, but then other gene expression studies where you have been done to support the equivalent circuits hypothesis model. So many people came to ask themselves, how can you use same types of data or gene expression patterns to come to these very different conclusions? And the issue using a slide from Georg Striedter is that simply that there is no simple one-to-one mapping between the expression of a gene and the function of that gene at the different layers of organization of the brain. And the brain is so complex and so difficult to understand because you can look at the brain and brain complexity at all these different layers, and genes contribute in different ways to development, but then they're used again to specify neuronal identity, to assemble circuits, and ultimately the activity of the circuits is what generates behavior. And to make this even more complex, natural selection, selective processes at the end are paying attention. Let's say, their selection is acting on the output of neural circuit activities, on computations executed by the brains in the context of behavior. But the selection ultimately through this very complex mapping is operating on the mutations that arise randomly in populations in certain species. So it's very hard by looking at differences in gene expression to understand how selection operated to generate these brains that ultimately are adapted in some way to their environment and different ecological niches that all these different vertebrates are inhabiting. And so I think molecular data should be used to link all these different levels of complexity in organization, and not just to look at one. And to do this, to make these links, we need to bridge the gap between developmental studies and studies of neuronal connectivity. The reason why this is useful is because if we ultimately want to use these evolutionary comparisons to understand something more general about how the brain works, systems from a systems neuroscience perspective, then it is important to bridge to go beyond the analysis of developmental stages and bridge the gap all the way to brain connectivity and function. And so this is where the analysis of cell types comes handy as a middle point for bridging that gap. And if you go back now to think about genes and how do we interpret gene expression patterns, how do we interpret what genes are doing, and we know that different gene batteries are used along all these different steps during development. But what makes this extremely complicated is that the same genes can be reused in different ways, and different combinations, from the patterning of the nervous system or to the specification and differentiation of different types of neurons, all the way to establishment of connectivity. So one way that I personally find useful to think about these problems is by drawing analogies with the hourglass model for vertebrate evolution, which was initially proposed by Denis Duboul when he discovered the collinearity of OX genes. And in recent transcriptomics work, Naoki Irie and colleagues emphasized the fact that this hourglass model of development can relates to the fact that at mid-embryogenesis stages, where there is a higher conservation of vertebrate body plans, there is also high pleiotropy of genes. What that means is that there are gene networks that are highly interconnected across organs. And what that implies is that if some of the genes in these gene networks mutate, then the entire development of the entire embryo is affected. So that means that there is a very strong developmental constraint, that this pleiotropy imposes a very strong developmental constraints that then ensures that this body plan does not change much across evolution. And what happens instead when you look at late embryogenesis is that the pleiotropy of gene expression decreases. So that means is that there are multiple molecular networks that become disconnected, and these are the gene regulatory programs that further specify the different organs of the body. And so one way to think about, going back to brains, one way to think about the problem of how do we interpret the gene expression data is that perhaps there is, in the brain, there's a similar situation where in mid-developmental stages, there is a very high pleiotropy in the moment where these brain regions that Loreta nicely introduced are specified. But then once the main brain regions in the vertebrate brain are specified, then and these molecular networks become more individuated and independent from each other. This is an opportunity for diversification and for the evolution of different new, perhaps, types of neurons in different species. And at the end, this divergence in neuronal identities is what supports also or underlies the evolution of different circuits that underlie the adaptation of animals to different environments and species-specific behaviors. So, to summarize this part, essentially, if we want to use molecular data to bridge these different levels of comparison, we need, first of all, to have a systematic way to compare neuron types across species, and to know what neuron types are more similar to each other and which are the new or divergent ones. But then it will also be important to link this, to understand how these diverse neuron types emerge from variations of development in order to link the analysis of neuron types in the adult brain to their developmental origins. And on the other side, to understand how these patterns of gene expression instruct connectivity. Alluding to what Luiz was saying earlier, one of the hardest things at the moment is to make, which I think we'll say is impossible, to make predictions about connectivity or to find correlations between gene expression and connectivity. I think at the moment we're still not there, and it will be very exciting to understand how variation of neuronal identity can underlie the evolution of neuronal connectivity. And I think this, with the scale and the depth of transcriptomics resolution, we're just at the beginning of all of this, and there is a lot of work that still needs to be done, both at the level of data collection, but also at the level of thinking about conceptual and analytical frameworks for integrating this data. And on the cell type front, we've done some work of this in the last 5 to 7 years by comparing differentiated neurons in the pallium of sauropsids, so in a lizard and a turtle, and more recently in my lab in the pallium of salamanders. And what we've essentially done is to take this problem that is a classical comparative neuroanatomy problem and to transform it into a comparative single-cell RNA sequencing problem. And for those of you that are not familiar with the pipeline, the way this works is that you take a brain section, you create a single-cell suspension, and then there are microfluidics methods to capture these single cells and barcode the messenger RNA in each individual single cell. Then everything is sequenced in a library, generating data that are huge data matrices where for each single cell you get expression value for each gene in the genome. And then these high-dimensional data sets are further processed to identify clusters of single cells. And the clusters are our proxy for cell type. And this is something that has become relatively routine for the analysis, for example, of mouse and human tissues. And what makes the work on non-traditional model systems a bit more challenging is the fact that with the single-cell analysis, we obtain single-cell suspensions and we reduce the brain into a soup. So we lose the spatial coordinates of these cells, and to reconstruct where these cells are coming from in the brain, it's necessary to go back and do in situ for marker genes. But the nice thing about having a single-cell data set is that then you can select marker genes for their cell type specificity instead of having to select them by doing drawing inferences from the literature. And another challenging part, which is still in a way under development in the field as a whole, is to learn and understand how to do these cross-species comparisons. So how to take these large data sets and extract meaningful information when we compare species that diverged even more than 300 million years ago. So as I said, we have collected all these different single-cell data sets, and most of this work is either published or in a preprint version. So I don't want to take more time to discuss the details. There's just one thing I would like to highlight. And so just to specify again, how do we compare this data across species? One of the things we can do is to use a computational pipeline that is called data integration. So this is schematized here. What we essentially do is to take all these different single-cell data sets, and we identify groups of genes that have similar patterns, transcriptomic patterns, across these different species. So genes that have a shared covariance across the species that we are analyzing. So this selection of genes is done in a completely unbiased way. We don't even know what these genes are. We don't need to know what these genes are in order to make these alignments. We don't pre-select them by knowing their identity, but we select these genes because after our computational analysis, we see that these are the genes that have consistent covariation patterns across these species. So they are the best genes that represent similarities across these data sets. And then after selecting these genes, we can take these complex data sets and bring them in the same coordinate system, if you will, so in the same high-dimensional space. And here we can define new clusters. And now these clusters are the clusters of cells that have highest similarity among the species that we are considering. So for example, some of the clusters will include only cells from one of the species. So these are species-specific clusters, indicating that these cell types are not similar to anything else in other species. But then some of the clusters will include cells that come from all the species that we have analyzed. So this means that these cell types, these clusters, are highly similar across these species. Doesn't necessarily mean that they're homologous, because the similarity may also be the result of convergent evolution. That's something important to keep in mind. And so we've done this work recently to compare the salamander to lizard and turtle. And what I'm going to show you now is this two-dimensional representation of the data from these three different species, color-coded by along this key that you see on the left. And so the first thing you see when you see this representation is that there is a relatively good mixing of cells from the different species, which indicates that there are genes, even in the adult differentiated brains, that are expressed with similar patterns across these species. And these are the genes that drive the integration of these data. And the major split here is between glutamatergic and GABAergic. That also makes sense biologically. And if we now focus on the glutamatergic cells, and now instead of color coding these cells according to species, we color code these cells according to brain area, which we have reconstructed with all the in situs and so on. What you can see is that there is in general co-clustering of cells from the medial pallium in salamander with cells from the medial cortex of reptiles, and cells from the dorsal pallium of salamander with cells from the dorsal cortex of reptiles. And interestingly, we also see co-clustering of cells from the anterior DVR of reptiles with cells of the so-called ventral pallium of salamander, which I'm to make things easier, I'm representing here with this color scheme. So essentially, what this data is saying is that there are genes, that using these genes that have maximum covariation across these data sets, the DVR of reptiles is more similar to the ventral pallium of salamander than to the dorsal pallium of the amphibian, which supports further the idea that the DVR and the dorsal pallium or dorsal cortex have two separate evolutionary histories, but it also enables us to trace back a little bit the evolution of the DVR to the tetrapod ancestor. And so something I want to emphasize is that these are this co-alignment does not mean that the cells are identical. There are many differences in these cell types. These are distinct cell types, and these differences in gene expression are correlated with differences in connectivity. For example, in the salamander, the ventral pallium is primarily the part of the pallium that receives olfactory inputs, together with this other green part, which is, we can discuss that later. So, in other words, with this analysis, we can extract sets of genes that probably are like the reflection in the adult transcriptomes of the developmental and evolutionary past of these brain areas. And with this specific example that I've highlighted, they give us some hints on the evolution of the DVR, supporting the idea that Striedter and Northcutt have nicely illustrated in this diagram. And one very last thing is this situation with dorsal cortex and neocortex is way more complicated. And one of the conclusions that come out from this transcriptomics data is that the neocortical pyramidal neurons are something very different, fundamentally different from pyramidal neurons of the sauropsid dorsal cortex. And this is best illustrated with a very simple diagram, where here I'm showing you the genes that have been traditionally used in the mouse literature to annotate the different layers of the mammalian neocortex. So you see, for example, that these genes up here are expressed at high levels in layer 2, 3, and then there are genes down here that are expressed at high levels in layer 6A. And so if just by going with the candidate gene approach, one may ask, okay, where are these genes expressed in the dorsal cortex of the turtle? Can we identify also by using these genes a layer 2, 3 cell or a layer 5 cell in the turtle? And the answer is that these genes are expressed either everywhere or nowhere. Or in other words, many of these genes, and most of them are transcription factors, that specify and maintain neuronal identity in the neocortex of mammals, are expressed in different ways and different cell type specific combinations in reptiles. So this suggests that even if these brain areas as a field, going back to the field homology concept, may be homologous because they are in the same position in the embryo, the cellular diversity that arises from these fields is incredibly different, and these cells are not even comparable at the transcriptomics level. And so the model for this would be that if there was that cellular diversity has evolved from ancestral cell types through this remixing and matching and re-elaboration of the regulatory networks that specify neuronal identity. And I think this was my last slide. So just to summarize, we're trying to compare types across species. There's a lot more to be done because, of course, the more species we bring in, the more we learn about brain evolution, and just there's only this much that can be said from the comparison of 2 or 3 species, and then there will be in the future, of course, more work from the community that needs to be done to integrate this with data from development and data from connectivity. And I will stop my presentation now if I find. Okay.
Thank you, Maria. This was very nice, very interesting. We have several questions. We have a very short question by John Kubi. Neocortex is a novel trait, really something new, correct?
In terms of
start.
Sorry?
We can start with that one because he just posted it in the chat.
In terms of cellular diversity, I will say so. So we have looked at this more in detail in this salamander preprint that the SOM bioarchive, where we have integrated together the mammalian data and salamander and reptilian data. And so we have reanalyzed essentially all the previous data from turtles and lizards. And the advantage of this new analysis is that we could use new data from the Allen Institute, where in the meantime they've done the entire cortical sheet, except the piriform cortex, unfortunately. So they've taken everything from the entorhinal to the neocortex, hippocampus. And so what comes out is that the salamander dorsal pallium has neuron types that at a very broad level are comparable to the entorhinal and to subicrum, but not really to the neocortex. If you do this data integration, you see that the cell types from the neocortex are in one place and the cell types from the salamander are in some other place. And this seems largely true also for reptiles, although I need to say that the majority of data from reptiles we have in this analysis are from lizards, because within the data, the turtle data set is by now very small. And we had more data for lizards, so maybe we're missing something because we don't have enough data from turtles, relating to the point we discussed earlier that the dorsal pallium in turtles seems to be, dorsal cortex in turtles much bigger. But we see that in addition, in reptiles, there is this cell type that is the ROR beta thalamo-recipient cell that is not present in the salamander. So our way of thinking about this is that there's been probably a stepwise complexification of the dorsal pallium or dorsal cortex, but that unless the salamander case is a secondary loss, which is something we can also discuss, probably the ancestral amniotes, at least, had a repertoire of cell types in the dorsal pallium, dorsal cortex that was more comparable to the entorhinal and to the subiculum than to the classical neocortical types that you find in mammals.
So, Paul, Luiz, Loreta, do you want to comment?
Actually, I wanted to jump in there a little bit regarding the especially the salamander paper from Maria's lab. I'm wondering whether those results, the sort of apparent absence of something really looking like mammalian cortex, does that effectively undermine the proposal by the Grillner lab that in lamprey there is something like dorsal Pallium? They have those studies, Priya Narayana, showing remarkable similarity in the lamprey. But if there's nothing like that in salamanders and intervening species, then that suggests it's convergent.
Paul, your micro is not working well. I think is it? Can you hear Paul properly? The others, because for me the sound is not great.
I think Paul's question was about what are the implications of these salamander data for the lamprey data that were discussed in earlier chapters. So, I mean, I can answer first, and then maybe also Luiz or Loreta want to comment later. But so there's two things. One is we cannot exclude fully that in amphibians or even salamanders there's been loss of something. So to really have a complete picture, one we need to go and look at more species. This is of course a never-ending endeavor, right? However, there is a very nice review by Georg Striedter, which is partly also covered in the book, but is more elaborated in the review, which enumerates a number of reasons why. The hypothesis that the dorsal pallium may have evolved either independently in fish and in tetrapods or even amniotes. So it's not entirely clear at this point whether what we call dorsal pallium in amniotes and tetrapods is the same thing or not. So that's one point. The other point that I would like to make is that the Grillner data are entirely based on physiology and connectivity, but there is no molecular data yet. There is a very nice preprint from the Eric Kessman's lab on the lamprey single-cell transcriptome of the entire brain. But so far, it was not clear to me whether the analysis of the single-cell data from the pallium is sufficient to really say is linking this molecular data to the Grillner data. I think there is not a clear link between the Grillner data and this transcriptomics data, and ultimately will be necessary to establish that link in order to compare this data in a fair way, because otherwise it would be unfair to compare molecular data from salamander with connectivity data from lampreys.
Loreta, you want to add something?
No, not much to what I totally agree with what Maria said. I know that there are a number of studies trying to find every single division in the lamprey brain, in particular in the pallium, and even things like the pyramidal tract and everything. And I think there is a point where I don't find that it is going a little bit too far. And as Maria said, that perhaps there is an independent evolution there of some parts of the pallium, but a different thing is that a dorsal pallium as we consider it in amniotes. Perhaps this is a different type of pallium. I mean, but not a dorsal pallium. So, yeah, this is what I think.
Okay, we had a a question by Luiz. I think we can invite Luiz on stage, unless you guys want to
say something. I have looked at with very detail at the data from Greener. I have really examined them very deeply. I don't agree with all aspects of the interpretation. I think that the data are strong. I think that the data, the results they obtain are quite strong scientifically, so they need to be taken seriously. I mean, they are not easily forgotten data, so they are there. They found this. Now, why did they found this, and what can be the explanations? There, I may differ slightly from the Greener's group interpretation, because I think there's a possibility that the data point into some form of my favorite idea, recent idea about the ring model of the cortex. So I was trying to interpret whether this data, instead of just looking for the neocortex like that in a lamprey, whether we should not first look for the entire pattern of the pallium, of the cortical pallium, and try to find an allo part and a meso part and a neo part, if ever it exists. Maybe one of them can be very small, diminuted, or not exist at all. So, in any of these primordial sort of cortical patterns that might have evolved in Agnata, they may have developed on their own independently, let's say by parallel evolution, functional properties and connectivity properties, because these things can occur in parallel. That means that being connected with the thalamus or being visual or being motor, that's not a thing that is written down in the genome in an absolute way. This can occur by intercombination of many different pathways of genomic influences. So I think it's very hard to do comparative anatomy only based on connections. As I have already recently discussed this point, so I'm not going to cover it again, but because I feel that connections introduces too many variables. So you go to proteins and protein interaction, protein interaction, and so it gets terribly complex just to see what is happening in your system. And when you are dealing with whole genomes or with all the genes expressed in the pallium, like Maria is and so on, that introduces a lot of difficulties that cannot be solved with the methodology we have right now. So I remember when I first met Manuel Molano in our first meeting, that the nature of my talk was the so-called ants country. You remember that, Manuel? Where I spoke of the idea that in biology, particularly, things tend to get very complicated before they become final and unitary. So that this sort of complexity intrinsic in when you deal with a lot of molecules interacting one with another has to be kept in mind. Plus the additional problem that we have to be careful with our assumptions. For instance, in the case of Maria's study, as I see from her images, she has been using largely the tetrapartite Pallium model, no? But that is an assumption for you. So since you are not personally involved in finding these models, you are using one. But what happens if that of Loreta is the right one? How would your data look if you use Loreta's model? Or what would happen if you use the ring model instead of the tetrapartite? So we cannot forget the model, the idea of a model, because when we approach a complex set of data, we need to have some sort of assumptions, which may be reasonable or not, but we attempt them to be reasonable, and that is conditions a lot the conclusions we reach and the meaning that we can extract from our analysis. So I recommend that people in this field be very conscious of which assumptions they are taking when they approach the analysis of the data, and also even the methodology. When you say, I go into the pallium, I dissect these pieces to isolate the cells. Okay, I will ask, show me this, how you dissect these pieces. Where are you cutting? What are you doing? Because that usually you just say that you dissected the pallium into this and that, but you don't really prove that you did what you thought you were doing.
So
we've seen from some other person, expert would say, well, maybe there your dissection was not as fine as it should have been. That's a methodological problem that we all have, all of us in our own field. So that you have to be conscious of this difficulty also. So
No, absolutely. If I may answer to this to clarify, because I understand that this data set generation and analysis pipelines can be very complex, and probably it's very hard to explain everything in a few minutes. So we are not using the tetrapartite model at any point of our analysis. So the UMAP, the plot that I've shown, there is an echo. Maybe Luiz, can you mute yourself? Yeah, better. Okay, thank you. So we're not using the tetrapartite model or any model at any point of our analysis. Our analysis is completely unbiased. And when I color code these dots, this map by brain region, I'm using brain regions that are defined in classical neuroanatomy literature. And I'm using these names, which unfortunately are names that we are carrying on from the different interpretations that people have have overlaid or used. So there is a reason why in amphibians the lateral pallium is called lateral pallium, but we know very well that the part of the brain that is labeled lateral pallium in the anatomy textbooks does not correspond to the new interpretation of the interpretation a few years ago of the lateral pallium. The models are changing, so it's hard to keep up. So, but for us, it made no sense to introduce a new nomenclature for brain regions, because that would have been even more confusing. So we're using these to relate what the single cell transcriptomes correspond to in space, but we're not. The analysis is not driven by that. It's only for us an illustration of our results, but all the clustering and the integration is not using in any way any information about where these cells are in the brain, is not using the tetrapartite or any other model. And again, we're not using the tetrapartite model, but we're only using the names of the brain regions as they became established in the literature. This is something I wanted to clarify.
I would suggest that beware of this idea that you are not using any model, because all scientists use a model. You cannot do without that. So if you say you don't use any, means that you don't know which one you are using. That's what it means.
No, it means that we're following the data.
You are using a model necessarily. So you can use the first one, then the second, then the third, then the fourth. But when you cannot do this, I don't use any one, because then you cannot do anything. In order to do anything, you need a model. So you are using a model for sure, absolutely for sure. Only so if you are not clear about that, then think about it. Which is my model? Which is the model I am using actually? Because you are using one for sure. I can't tell you.
Maybe we can let Luiz ask his question.
The axis of the brain. What is rostral? What is caudal? You are not using any idea about that? What is rostral? What is caudal? What is dorsal? What is ventral?
Well, we are sequencing cells, so there's no place to introduce this information in our sequencing data.
As a scientist, you must have evolved a lot of models. Some of them you are conscious of them, maybe some of them you are not conscious. That's normal. That happens to everybody. That some of our assumptions we are not conscious of them as being possibly problematic. When we approach novel sort of data, particularly, that's the moment when the new or old ideas really become critical.
Okay, let's Luiz ask his question, because he has been waiting for some time. You have to unmute yourself. Well, I can unmute you.
Hear me?
Yes.
Yeah. So thanks for inviting me. Yeah, the question that I had is actually for everyone. And I think it goes back to something that I really like that Maria brought up, which I don't see discussed. I mean, I obviously I don't work in this area, and I read some of it, but I don't seem to see it discussed as often, which is a desire to find these almost one-to-one mappings across these diverse 500 million years of evolution and the difficulties that this poses. And I asked this question in the context of the structure that some of us are really interested in, the amygdala, which is a really tricky one because it really straddles a division that is the Pallium and the subpallium, and the difficulty of understanding its organization in reptiles and birds, not to mention others, and amphibians and fishes. So I guess my question is twofold. One is, are we a little bit closer to understanding what might be something like the paleoamygdala in reptiles and birds, or are we really still very far? And also, do we need to promote a little bit more strongly to the community of neuroscientists in general that even though we talk about lots of structures in the brain as highly conserved, it doesn't mean that we actually can point or know exactly where these things are across vertebrates? Because if you ask a general neuroscientist would believe that, of course we must know where the amygdala is in fishes and amphibians and so on and so forth, because it's so well known that it's so well conserved. So that's what I wanted to hear a little bit, thoughts from the panel, from everyone, to see what directions are fruitful for the field to focus on, to understand, to research, and to communicate.
Can I say something? Yes. Yes, Luis, this is a very important questions. I could hear only the connection was not very good, but I think I could follow some of the questions that you raised. One of the things that is that we are proposing homologies at field level, and these fields include some cell populations that might be homologous, but not others. And there's also some, a lot of variations. There is a lot of divergence. So perhaps trying to find one-to-one homologies in all cases, perhaps it doesn't make sense, no? Perhaps. So I know that some areas, I'm one of the ones that is always arguing that in some parts of the Pallium you have the paleoamygdala, and it has these features that are similar to those of the paleoamygdala in mammals. But the paleoamygdala in mammals, you have many subdivisions of the lateral amygdala, many subdivisions of the basolateral, many of the basomedial, a number of cortical areas. So there, and in each of those groups, even you have different cell types, different glutamatergic cell types. So it's tremendously complex. It is also so for the subpallial for example, for the subpallial we have been trying to identify specific cell types coming from the same embryonic divisions, having the same molecular profile, or at least partially, because we don't have all the data, like Maria, for example, has when she's trying to do this single cell transcriptomics. So it is very complex. We only play with a limited amount of data, and due to the huge divergence, perhaps it doesn't make sense to go to the one-to-one homology nucleus by nucleus. Perhaps we should go to cell populations with the same embryonic origin, and perhaps sharing some other similar features, but perhaps we don't have a high degree of conservation in those populations that we propose. We had only partial conservation, and even we might have populations which are completely new, and because of the high divergence that we have. So I don't know, this is, and if we should be making more pressure in the community to change the chip for example, prefrontal cortex or neocortex, versus saying that this is more similar to the amygdala in general, it is very difficult, a very difficult project, because we have many researchers in the field, especially in the USA, following something more similar to the Carten's hypothesis still today. People that do not follow really, or do not understand perhaps what it means, the topology, no, the topological position, they go more in topography, and topographic relationships change a lot because of the deformation of the tissue. I think that many people do not really understand deeply what the topology means, and the importance of this for trying to achieve a better understanding of the divisions and the embryonic divisions and the populations that are generated in each division. So it's difficult. I'm not going to try to convince anyone out there. I know that many people will not agree with the things that usually I'm proposing in the articles, or those that Luiz proposed, or so. We even have disagreements, little disagreements, Luiz, between us. But globally, I think that we had a general view of looking at the data, and I think that I can be more in agreement with Luiz's ideas, and I think that I'm sure that Maria also is in this type of view. But many other people follow a different type of arguments, and it's going to be difficult to try to convince everyone. I think that at the end, in the long range, perhaps more people will come to understanding the brain as we try to do. The other day, by the way, I was reading an article, Luiz, I was reading an article by Martin Weil from New Zealand. And usually, this is a very important researcher. I admire his work a lot. He studied a lot of connectivity patterns, and he has done an enormous amount of work there in using the pigeon, mainly the pigeon as a model, but not only, other birds as well. Usually he has been defending the idea that the DVR is more like the neocortex in many of his papers. But suddenly I was surprised that 2 years or 3 years ago, suddenly he was studying the connections of the ventromedial hypothalamic nucleus, and he found a group of cells in the caudal nidopallium, and suddenly he realized that it was exactly the same that Fernando Martinez Garcia and Laura, Bruce and company were defending a long time ago, when they say this is like the Pallium amygdala, and the Pallium amygdala parts that project to the ventromedial hypothalamus. So I was surprised finding in the discussion of his paper something like that. Perhaps the caudal nidopallium is like the amygdala. So perhaps at the end, in many years, people little by little will realize that some interpretations make more sense than others. But it is okay also if some people want to make emphasis on analogies, as soon as they recognize that they are talking about analogies. Analogies are very interesting. Convergence is extremely interesting. Convergence to achieving a particular function, for example, in 2 different structures, that they converge to achieve something. Perhaps by studying that in detail, can tell you minimal requirements that are needed to achieve that function, and that is also extremely interesting. It is interesting to know, to understand how this convergence was achieved. So, well, everything is interesting, convergence, divergence, but I think it is very important to know what we are talking about. If we are talking about homologies, or if we are not talking about homologies. It is very important to distinguish the situation, because at some level, it will help you to understand better what you have there, and to address better questions, and to make better predictions. If you
So I don't know if maybe we should move on. I don't know if the other panelists want to add anything, or it is already 8 p.m. here in Spain, so the 2 hours we had booked are elapsed. But if you want to comment, or we can even invite some other people that would ask questions, but it's of course up to you. We can also leave it here, or just
maybe let me say as my last word in this, that we have to be very cautious with the idea that homology is similarity, because homology does not need similarity at all. You can be similar and homologous, or completely dissimilar and homologous. So it has nothing to do with this feature. Homology is sameness. So what do we mean with sameness? That's a big interrogant. So how can be distinguished logically sameness from similarity? That's the point. And that's the point that has not been resolved in the field. That's the problem.
Paul?
Yeah, I would add to that many of us really care about a particular lineage. Like, for example, want to understand humans, and therefore mammals, and therefore amniotes, etc. And so for us, the issue, I'm speaking for myself here. The issue of homology is primarily about what do the structures in other lineages tell us about this lineage of interest. And so, birds are fascinating, but given the high degree of independent evolution, perhaps they don't tell us so much about our common ancestors, and we should be really focusing all of our attention on the tuatara, or some reptile that perhaps better represents the common ancestor that we have. And, Drosophila are fascinating, but they're really very different. And so, they're interesting. And convergent evolution is very interesting to understand mechanisms. But if you want to know the specific mechanisms of a human, then there's some things about Drosophila and birds and turtles that are not really going to give us that many hints. And I think that's where the problem lies. Too many synapsids have died, and too many, non-vertebrate chordates have died. The real problem is all the extinctions, I think. And I think that the thing that always excites me about homology, and especially sort of the developmental homologies, is that maybe that is something that gives us the hint about what the common ancestor had, even if the mechanisms that we can study in extant species are really, really derived. That's the challenge.
Maria, would you like to add last comment?
No, just building on what Paul said, the other limitation we still have is that our phylogenetic sampling is still very limited, even within the sauropsids. We're looking at one crocodile and one turtle and a few lizards. So there is variation even within these different clades, and it is very important to expand our range. It would be super interesting to see more data from more fish, not just teleosts. More data from sharks, for example. Going back to Paul's question on lampreys, this to fill all these gaps, even just collecting more high quality data from these animals, or from lobe-finned fishes, it would be extremely insightful. So there's definitely a lot more to do.
Okay, so maybe, unless you want to add something, we can leave it here.
Yeah, I'd like to actually add, could we establish a funding body that directly supports filling those gaps. In other words, write a grant about sharks and you get a good shot at it. But if you're writing it, about, in other words, really target the species that from a phylogenetic standpoint, we can say are going to be really good ones to invest transcriptomics, connectivity, optogenetics, physiology, everything that people have applied to sort of sparsely sampled species, and actually target those gaps deliberately, given our current assumptions about which ones are the right species. I think that would be fantastic. If I had $1 billion, that's what I would do with it. I'd give it to people doing sharks and placaras and whatever.
Well, you're ending the discussion on a pessimistic note, because you know that in terms of funding, things can be very difficult with this kind of work. But
okay, so we will leave it here. Sorry for all the questions that we didn't answer, especially John Kubi, because actually he made many questions. There are some in the chat and some in the Ask a Question button, but it is already a bit late for everyone. So yeah, I would like just to thank Raquel and Alex, who also always help in the background, and of course the speakers, Loreta and Maria, for the nice, wonderful talks, and all of you for your questions. And I don't forget Luiz and Paul, who are always here, of course. Okay, I will cut here. Thank you. Bye.